Hi, my name is JC Noble, and I decided to learn a bit more about the article titled "A Newly Discovered Cell Helps Pythons Poop Out the Bones of Their Prey." Super fun stuff. But to give a bit of a background, snakes are well known for swallowing their prey whole. So this would include mice or other small vertebrates, and they are also what's known as intermittent feeders. So what this means is after eating, they fast for weeks or even months at a time. And during this fasting, the secretion of digestive juices stops and the lining of their intestines atrophies, meaning it shrinks. This also includes the cells with the finger-like villi that absorb all the nutrients. But as soon as a prey is caught, this digestion starts back up again, and the intestines and the villi enlarge in order to accommodate all those nutrients that are about to be absorbed, as well as the stomach's pH that was nearly neutral at the time of fasting drops to a pH of 2, making it very acidic and perfect for breaking down the vertebrae and prey that just entered. So, with this method of swallowing their prey whole, it means that they are digesting the skeleton as well. And with this particular diet, it results in high influxes of calcium and phosphorus during these feeding times. So, this led researchers to question how are the prey's bones broken down, and how do Burmese pythons specifically control the amount of calcium hitting their systems during feeding time? So, in order to answer this question, they implemented 3 different diets. This included a normal diet, which included the entire rodent, skeleton and all, a low calcium and phosphorus diet with boneless rodents, and then a calcium-rich diet with boneless rodents that were then injected with calcium carbonate supplements, just so they would know exactly how much calcium each snake was getting. The effects were then analyzed using light and electron microscopy techniques, specifically alizarin red S and peroxidase staining, as well as EDX analysis. Also, they tracked blood calcium and hormone levels. This then revealed to them the presence of specialized cells that collects bits of calcium and phosphorus around an iron core. So what's above me here is a transmission electron microscopy image that shows, sorry, it's mirrored, the finger-like villi here we see kind of has this pit, has this pit here that is actually a multi-layered particle of calcium and phosphorus as a result of the broken down bones. So they noticed that when the snakes were fed a calcium-rich rat, they observed this here, showing that the cell was collecting those excess dissolved calcium and phosphorus and kind of making bits. And they had a good example in the article where they compared it to a particle of dust that collects ice to form a snowflake. So, a good way to think about it. And then, of course, when snakes were fed the boneless prey, so uh, not as much, or actually no calcium, or not no, but a lot less calcium, particles were not produced by this cell type. And although there were iron elements that were located within this crypt here, it was empty. So this kind of tells us that when the snakes are fasting, these crypts here remain empty. And then when calcium enters the system, the excess is then collected here. And this kind of pellet here is excreted through the feces. So this answered the question of how they control their calcium and phosphorus intake when they absorb this entire skeleton. So they also found the cell type in other snake species and even other reptiles that eat vertebrates, such as the Gila monsters, that huge snake. And for future research hopes to look at certain species of vultures that are also known for ingesting the whole prey. So why I chose this article being on the pre-vet track, I'm interested in animals, love animals. So when I saw the snake, I immediately was intrigued. And then also in the title, it said "Do Discoveries," which are always interesting to me. I also love this article because it made many connections to cell biology, which included cell specialization and function, along with also microscopy and cell imaging, which we learned a little bit about in our first lab. So, learning about these specialized cells and learning about the Burmese pythons and their processes, it got me thinking about humans and led me to ask the question, how do human intestinal epithelial cells compare to a Burmese python specialized cells at absorbing calcium? So, thank you so much for taking the time to listen, and here are my references. Thank you.